Hello everyone, and we are going to start our next lecture series with Chapter 7, and I'm going to use a little bit of a different technique, and I'm going to try out my little tablet and this new pencil thing I have. So we'll see what we think of it, because I really want to try some drawings as we go through the nervous system. So here are our objectives for Chapter 7. It's really a great intro, looking at the tissue, looking at the cells, the neurons, and of course the supporting cells, the neuroglia. So we did a nice little activity looking at nervous in particular, and we're really saving muscular for a little bit later. We do muscular pretty quick coming up. When you get time, I always like to put some links to a good overview video, usually by Bozeman or Crash Course or something like that. So after you've done the hard work, after you've listened to the lectures, these videos really do a nice job of summing things up. So I always recommend doing those after you've kind of done some of the work, because they talk really fast, and when you first watch them, it's a little overwhelming. But once you have studied this a little bit, you're like, oh, it's really starting to make sense. It's really starting to come together. So I like their videos. We talked about getting all of these terms, and you're going to see a ton of terms, a ton of synonyms, and it's okay. You just get used to it after a while as you study science, and you're like, oh my gosh, there's a name, 7 names for one structure, and by this time, maybe you just start rolling your eyes going, okay, what are all the names for this? And so they give you a nice little table showing you the different terms and what they can be used for, especially like when we're talking about like the action potential or something like that. You may see it abbreviated, you may see it as a spike, a nerve impulse, a conduction signal. So lots of different variations for just one term. So as we look at the organization of the nervous system, there are really 2 main parts. We have everything located in the center of the body that's really encased in your cranium and your vertebra, which is where your spinal cord is and your brain is housed. So this is the central nervous system. It's only composed of your brain and spinal cord, and it will have some special neurons that are inside and only found in the CNS. The peripheral nervous system is everything outside of the central nervous system. And this is where you see your big nerves. Lots of sensory nerves, lots of motor nerves, lots of mixed nerves. And it's color-coded in yellow on this diagram here. So anything outside of the central nervous system will be the peripheral. So the main structural and functional unit of the nervous system is the neuron cell. The neuron has some really special functions. It can respond to the world around you. If there's a change happening around you, whether it's external or internal, you will respond to that stimuli. The cell can pick it up if it has the right receptor. When it picks up that data, that information, it can conduct an impulse. It does this by generating electricity and chemical energy. It kind of flip-flops back and forth depending on the type of neuron and the types of synapses that we find. And we're going to talk more about synapses at the end of Chapter 7. They can use their chemicals to regulate things in the body. They can regulate our moods. They can regulate so many processes within the body. So if you get really excited, what happens to your heart rate, your breathing rate? A lot of different things rely on these chemicals, or you'll know them as neurotransmitters. And of course, one of the really cool functions about neurons is you're going to find them where your big 5 special senses are. We know we have more than 5 senses. We have everything from balance to proprioception to vibration. All of those, plus our special senses like vision and touch and taste. And so that's all your perception, perceiving the world around you. You guys did phenomenal in the on-ground class looking at neuron structure. So if you're in physiology, you've done an anatomy class, you've done something or have the background to be at this level. So you've studied a neuron, and most of you drew this type of neuron that you see at the bottom here. This is what we consider a motor neuron, and it's the most numerous type of neuron in the body. And you can see some really important parts that we are going to talk about. One of the most important parts is the dendrites. And it's going to be really important because their job is to send impulses to the cell body. And of course we have our cell body, and then we have our axon. And our axon is this really long process going through all those little myelin sheaths. The axon will carry the impulse away from the cell body. So think A for away. The other parts that we're going to look at, we're going to look at the myelin. We're going to look at myelin in the peripheral nervous system. We'll look at myelin in the central nervous system and its role and how it's created and why it's so important in maintaining neuron function. What we're going to wait to talk about until closer to the end of the chapter is this right here. There's so much that happens here. This is called the synapse. The synapse is a junction. This is where two cells are going to meet. And there's a lot that takes place at this synapse. So we're going to kind of save this special area until part 3 of our lecture with the nervous tissue. So this was just another really nice diagram that your book had that I wanted to put into your PowerPoint because it has a really a couple of more really important structures. The node of Ranvier. So when we talk about your axons being able to take an impulse away from the cell body, your neurons can run at 250 miles per hour. That's really fast. So they utilize this insulation. Wherever there's insulation or wherever you see that myelin, we don't have to run the electricity through it. The electricity can jump to the node and jump to the node and jump to the node and jump until it reaches the synapse, and then we can transmit the signal to another cell. That is absolutely phenomenal. We can just skip, and we go to the next cell. It's almost like cheating if you really think about it. Now, if we didn't have this myelin, we would have to run the entire length of the axon, and we would have to open up all the proteins, propagate the entire electrical gradient to get the impulse to go. So that's what my red arrow is showing. My red arrow is just saying, hey, if we didn't have these really cool myelinated regions, we would have to run the entire length of the axon. I would honestly rather just skip, and move on to the next one. So myelination is very, very important when we talk about functions of neurons. Now, neurons don't just transport electricity or neurotransmitters. They have to keep the cell alive itself. They have to do normal cell function as well. So this is called axonal transport. You have to remember that some of these cells break the rules of what a cell should be. We think of cells as itty-bitty, super microscopic, round little cells. Neurons do not look that way. They have these really crazy looking cell bodies. They have branches coming off of them. We see these really long branches, these axons. Axons can reach from your spinal cord in the middle of your back and go all the way down your leg. So you can have axons 3 to 4 feet long. That's crazy how long these cells can be. And we have to make sure we can maintain the entire length of that cell. So that's where axonal transport comes into play. Moving materials from your cell body, all of the proteins and everything that we're making here in our little factories, we need to be able to get it out and to different parts of the cell, including the very end of the axon. So we can make stuff in the cell body and ship it out away from the cell body and go this way, like so, and get nutrients and different building blocks out that way. Or maybe something is happening in the axon down here. Maybe we need some more organelles, more vesicles, like maybe we need some more supplies, or maybe we have too much of something down here. I'm getting ahead of myself a little bit. And we need to ship it back to the cell body, either to be broken down or something. And so this is called retro, going back towards the cell body. And we said this was really medically significant because this is how viruses can travel and infect cell bodies and live there the rest of their lives, because they can hitch a ride on this retrograde transport. As far as classifications of neurons, we have different ways we can look at the neurons. We can see their job and classify it that way. This is probably the most popular. This is how we really talk about neurons when we talk about them in everyday life. Motor, this is kind of like a special category because motor has a lot going on. So it's more of a special category. Or we can talk about structural. What does it look like? And we can categorize it that way. So we made a nice little table in class where we really just focused on the functional and then moved into the structural because they're actually really related that way. So let's look at the functional classifications first. As far as functional, it just means in what direction is the impulse going. For sensory neurons, we are taking information from the outside world and sending it to the central nervous system. So it goes out to the center. You'll also see these neurons known as something else. So this is kind of your AKA, also known as. And they like to call them afferent neurons. Think of A for arrive. So these are your arrival neurons. They're arriving with information that is happening in the body or around the body. And they're arriving to the brain, be like, hey, this is what's going on. We also have motor neurons, and motor neurons leave the center and go out into the body. And we're going to find usually a target organ of some kind, usually a muscle, maybe a gland. And since they exit the central nervous system, you see this nice E here, you'll see them termed as efferent neurons. A very special neuron you're going to find in the central nervous system only are association neurons, or they're also known as interneurons. And they just really help integrate, decide kind of what to do with the data coming from your sensory neurons, and they help send commands to motor neurons. You can see our motor classification down below. It's kind of our special classification. We have an area of what you can control, so that's where you see that voluntary control. It's really just your skeletal muscles that you can have control over. The other section is your autonomic. This is your autopilot. These are things you cannot control, so they're completely involuntary. This deals a lot with your smooth muscle. Think about where you find your smooth muscle. This is around a lot of your internal organs, your blood vessels, cardiac muscle. A lot of your glands will secrete things. You don't know when your pituitary gland is secreting hormones. You don't know when your adrenal gland is secreting hormones. So your autonomic nervous system takes care of that, and that's the motor part. And we talked about two subcategories of the autonomic motor: sympathetic versus parasympathetic. When your sympathetic nervous system is in control, you are in fight or flight. You have a lot of adrenaline pumping through your body, and your body's ready to go. It's primed. Parasympathetic is the opposite. We're rest and digest. We're really going to use those glands. We're going to get nutrients from our food. We're going to heal. It's a very chill part of the motor nervous system. So that's all with function. We can also classify neurons by the way they look. And by the way they look, we usually look at the number of branches or main processes that are extending from the cell body. The main ones that your book uses are pseudo-unipolar, false one pole. This is what your sensory neurons look like. Bipolar means we have two poles. This is also a sensory neuron, but it's a very special sensory neuron found in the eye. The rest have several branches off of the cell body. And this is what we normally draw when we draw a neuron. We draw the multipolar, all these branches, all these dendrites, one long axon. It is definitely the most common type of neuron. And as far as what type of functional neuron looks like this, your motor neurons, so motor neurons look like this, and your interneurons look like this. Your book has some nice pictures showing you kind of direction of impulse. Hey, here are my receptors. The direction of impulse is going this way. And I have this nice one pole, one process. The cell body's pushed to the side. So that's how I know it's a sensory neuron. It's pseudo-unipolar. And I just follow the direction. So these right here, this is a dendrite. And all of my dendrite branches here. And I go toward the cell body. When I go away from the cell body, that's the axon branch. And you can see things labeled the same way over here. They give you the arrows, which is really nice. We can see the arrow going to the cell body and the arrow going away. So anything going away is the axon. Anything going to the cell body, those are all the dendrites. As far as a full nerve, notice we've been saying neuron the whole time. A nerve is a bundle of neurons outside of the CNS. We can have an entire nerve made of sensory neurons or motor neurons, or we can see a mix of neurons in nerves. Nerves are actually pretty big, pretty thick. Now we can move into neuroglia. These are the supporting cells for your neurons. They outnumber neurons 10 to 1. So let's look at the supporting cells in the peripheral nervous system first, because there's only two. The most important one you need to know is the Schwann cell. Many people are familiar with it because they like to draw these Schwann cells on their motor neurons. And so you see these Schwann cells when students draw their little thing here. They'll have their little neuron, and then they will draw the Schwann cells on top. They'll label it myelin, which is fine because that's what the Schwann cell makes, but they also create this really fantastic structure called the neuralemma, and it helps protect and sheathe the myelin and the Schwann cell. So you can see it's like this outer coating right here. And it's going to become more important later on when we talk about the Schwann cell in more detail and regeneration. Satellite cells are just supporting cells that actually surround and protect the cell body. The CNS definitely has a lot more going on. We have very special oligodendrocytes. They're the cell that will form myelin sheaths for the central nervous system, in the brain, in the spinal cord. They form it way differently than the Schwann cell does in the peripheral nervous system. So first of all, they do not have this amazing structure, the neurolemma, which is a big bummer. And here you can see there's one oligodendrocyte. It looks kind of like this weird octopus thing, and it's going to wrap its tentacles around all these different nerves. And so one oligodendrocyte can wrap itself around several neurons, whereas a Schwann cell, like I had to draw 4 Schwann cells to help cover one axon of one neuron. So Schwann cells are much smaller and they're much more localized. Microglia help protect the CNS. They look like little tumbleweeds down here. So you can see them down here, and they help eat up foreign and used up material that you find in the central nervous system. Astrocytes are definitely the most popular and most well-studied neuroglia cell. They're going to help regulate the environment that's within the central nervous system. And they're named that way. You see the word astro because when they first started looking at them, they thought they looked more like stars. And then our last type of neuroglia is an ependymal cell. And you're going to find these in special areas in the brain. And their job is to actually make the fluid that your brain and spinal cord float in. And they make a lot of it. They make over 1/2 liter every single day. And that's quite a bit. There's not a lot of space for this fluid in the skull and the spinal cord. So they've got a big job. Let's look at that myelin a little bit closer. Myelinated axons conduct or help conduct impulses very, very quickly because it helps the impulse skip along the axon. When we start to lose myelin, it can be very devastating. And we have lots of conditions that show this, very famous conditions that a lot of us have heard of. So conditions that result in damage to the myelin, we see a multitude of symptoms depending on what neurons are actually affected. Some famous ones are multiple sclerosis, Guillain-Barré, Alzheimer's, even concussions. So you can see with some of these radiographs how, a lot of the areas of the brain just start to shrink away because they're just disintegrating basically. And this area here, this is where the hippocampus is located in the brain. And you can see in a normal one, it's very robust and very full. And here we have just a gap. All the neurons there are gone or demyelinated, and we cannot see them any longer. And the hippocampus is in charge of long-term memory. So that's really, really rough for those patients. Multiple sclerosis is an autoimmune disease that actually affects the myelin in the central nervous system. And so it's going to attack the oligodendrocytes. And you can start to see these almost like kind of like scar tissues start to form when these are attacked. Guillain-Barré is an autoimmune disease that will affect all the myelin in the peripheral nervous system. And so they're trying to show you some thickening down in here of some of the neurons going to the legs. So this patient probably has weakness in the legs and trouble walking. And then of course, one that is now finally being studied in especially professional athletes or even just serious athletes where they have lots of concussions. And so we start to see over time, if you have lots of brain injury or trauma, that can actually demyelinate the brain as well. So let's look at some healing of the neurons, since we've looked at some of those diseases, especially some of those diseases that will attack the myelin. So how do we, if we injure a neuron, how do we get it back? And can we get it back? Well, if you're in the peripheral nervous system and you cut some nerves, you're in luck. If you've cut the axon, so you can see the site of injury here, if you've cut that axon, we will be able to eat up and get rid of all the injured parts. And the Schwann cells can use their neurolemma and help form a regeneration tube. We can also start to form growth factors and can stimulate the growth of a new axon and reattach the axon like nothing ever happened. That's fantastic. Now, in the CNS, you're not likely to get any regeneration. So if you sever some neurons inside the spinal cord or the brain, we don't get that neuron function back. We are looking at the oligodendrocyte itself as the main culprit. They will actually produce even death receptors and make oligodendrocytes around them die. We see inhibitory proteins coming from oligodendrocytes and the other myelin sheaths to help prevent regeneration. And astrocytes even get in on the action and create scarring. So you can't form a regeneration tube and get that neuron to recover. So the CNS has a lot of things working against it as far as recovering. Another really interesting phenomena due to all of these neuroglia is the creation of the blood-brain barrier. It's a protective barrier, and it's created by astrocytes. Astrocytes will actually send out these little processes, and they call these like feet or podocytes is the real term for them, but they're actually little feet, and they will wrap around every single capillary and really like seal them and create these really, really tight junctions. So then it's really hard for things to get through the membrane. We just did a test over what can go through a membrane. So it's very, very selective. And it's selective unless you look like a membrane and you're fatty or you're small and uncharged and don't really need any help gliding through a membrane. That would be some fats, gases. We talked about general anesthesia, which is why they use a gas mask to put over you when you go out for surgery, when they put you out or put you to sleep. And even things like alcohol and nicotine can actually just zip through the entire barrier and get into the brain, which is why they can cause such quick effects. So I think we're going to stop there for our part 1, since that was our introduction to chapter 7, and then I will create a video just based on membrane potential that you see here, and then we'll look at the synapse and the other structures for our part 3.